Welcome to Bio class. Today we'll discuss here new chapter respiration in plant. We know all the organism will breathe. But why is breathing is so essential to life? What happens when we breathe? And uh, all the organism including plant also can breathe, microbe also can breathe. If so, then how? We have to understand that the energy to maintain their body, and the mechanism of the body, this energy it is produced in the cellular respiration. So for this energy, that respiration is required. Respiration always does not mean that oxygen is involved. I think you know that there are two different part is there, that is aerobic part and anaerobic part. But in aerobic respiration, the oxidative phosphorylation will happen, so more energy is produced. If we can see this part and can able to understand better. The cellular respiration, this is the cellular respiration. In cellular respiration process, what will happen? Here the food material, every cell will gather the food in form of glucose. In form of glucose, that food is gathered. Okay, food is reached uh, in the cell, every living cell, and after that, this food, the glucose splits. Glucose here the process of glycolysis. Lysis means it breaks. So glucose breaks in cytoplasm, inside the cellular cytoplasm, into 2-pyruvate. So glycolysis is the first process. Glycolysis is the process which takes place in cytoplasm. In cytoplasm, in every organism, glycolysis takes place, whether this organism use oxygen or not. When the 2-pyruvic acid or 2-pyruvate is produced, then this pyruvate will enter inside in the presence of oxygen. And here the citric acid cycle, or TCA cycle, tricarboxylic acid cycle, or Krebs cycles. Three names here. Three names are very popular according to Hans Krebs, German scientist. Hans Krebs, that name Krebs cycle, and the citric acid cycle. That is the second phase, and the electron transport system ETS inside the mitochondrion. Always remember, when oxygen available, then inside the mitochondrion, this oxidative phosphorylation takes place, and 36 molecule of ATP is produced from the one molecule of glucose. But when oxygen is not available, then cell goes for the anaerobic respiration. In case of the human, the muscle cell can do that anaerobic respiration. Otherwise, the fermentation, there is some separate organism which can do that. And here the same number of glucose can produce only 2 molecule of ATP in both the cases. So anaerobic respiration, it gives less amount of energy. So this is the basic, and here detail about the glycolysis is there, detail about anaerobic respiration, fermentation, citric acid cycle, and electron transfer system is there. First process here, glycolysis. Glycolysis, here the glucose, which is 6 carbon containing, glucose converts into glucose 6 phosphate in the phosphorylation, the help of hexokinase enzyme. Here ATP is produced. Glucose 6 phosphate converts into fructose 6 phosphate, and then fructose 1,6 bisphosphate or diphosphate. So here the phosphorylation will occur, isomerization and other all other thing. Triose phosphate, triose phosphate here the here glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. If you can recall, then all the name you have got in photosynthesis when the glucose is produced, and here the glucose, when it is breaking, it goes that the reverse route. Almost you will get on many name from here. Okay, so this is that in your book which is given, and here the ultimately that pyruvic acid will produced, that you can see that pyruvic acid, which is 2 molecule of the pyruvic acid, is produced. If we can see these things in different view, the glycolysis, only the view here, only to understand this one, the view here, the glucose we are writing a little bit elaborate form. So to understand many facts from here, this is the glucose, is the start point. So glucose converts into glucose 6 phosphate. Here the phosphorylation will occur. The sixth carbon of the glucose, the phosphorylation. So phosphorylation hexokinase the enzyme. Now this one, the isomerase, that the glucose 6 phosphate converts into fructose 6 phosphate. Look at here, the glucose, the aldehyde form, now it turns into ketose form. So it is fructose, and it is fructose 6 phosphate. Now fructose 6 phosphate converts here fructose 1,6 diphosphate. Here is the 1 carbon and the 6th carbon phosphate. Here the phosphate 1,6 diphosphate or bisphosphate. Here phosphofructokinase can convert this structure. Okay. Now this is 6 carbon. This 6 carbon it cleaved into 3 carbon form with the help of cleavage enzyme aldolase. If you can recall that in photosynthesis it was joined, now here it is separating. Now here dihydroxyacetone phosphate, here is the ketone group, dihydroxyacetone phosphate and glyceraldehyde 3 phosphates, glyceraldehyde 3 phosphate in 3 carbon phosphate and dihydroxyacetone phosphate. These two are isomers. So very easily that isomer can, triose phosphate isomerase can convert this one. If we can consider the glyceraldehyde 3 phosphate is there, so it turns into 2 molecule. So after that we will write this one, the 2 molecule. Okay. So next, if we see this part, look at here, that glyceraldehyde 3 phosphate, now it turns into 1,3-diphosphoglyceric acid. So here the phosphorylation will take place. Now it is 2 molecule, and look at here, nicotinamide adenine dinucleotide, it takes a hydrogen from here in a DH2. Okay. So so diphospho 1,3 diphosphoglyceric acid, it converts into 3 phosphoglyceric acid by removing 1 phosphate here. Here only 3 phosphoglyceric acid. 3 phosphoglyceric acid with the help of that mutase enzyme, the phosphoglyceromutase, which the 1 molecule of water will move out, HOH will move out, it turns into phosphoenol pyruvic acid. From here, actually this is the phosphoenol pyruvic acid, and this is 2 phosphoglyceric acid. Now phosphoenol pyruvic acid, it can convert by removing the water. Okay, this phosphoenol pyruvic acid now turns into the pyruvic acid, 2 molecular pyruvic acid. Okay, so in your book whatever there, and this is no difference, but only that in structural formula that I have shown to understand that exactly where the phosphate is the, which carbon phosphate is the, how the removing of all this, how the cleavage will take place, all everything. All right, so only for the understand. And now here in uh, this pyruvic acid, here you can see that the enzymes which is involved if you are interested, but this much detail it is not uh, in your book. Okay, now in these there are already it is told that there are two way that pyruvic acid can uh, I mean in anaerobic do the anaerobic respiration. So in our muscle, that lactic acid, so it turns into lactic acid by removing that the hydrogen. Okay, and it also turns into ethyl alcohol in case of fermentation. In fermentation, and this is in alcoholic fermentation, yeast and bacteria, it turns into ethyl alcohol. So here that in ADPH2 the supply that hydrogen. So that is about here the glycolysis, this process, whole process you can see. After glycolysis, that next it is there that uh, in aerobic form the citric acid cycle, Krebs cycle and electron transfer. So next part we will discuss citric acid cycle and electron transport system. Thank you.